Hi, I want to say good morning, good afternoon, or good evening, but it depends where you are in the world. But let me jump straight into this. This is the weekly ZP Developer Zone webinar. As you can see, as per normal, I've got some kit in front of me. I'll probably talk up a little bit about wearables today. I'll definitely talk about screen printed electrodes. I'll talk about hydrogen peroxide. I'll talk about lactate. And all sorts of things. So let me just jump in forward a little bit and actually just give you a quick heads up. So today's I want to say it's 8th of December, but in fact it's the 7th of December. It's sometimes easier just to go a bit earlier. Just to say, if you buy off the web store, you can use this discount code. But if you don't buy off the web store, we can't give the discount code because basically the cost of administration then makes it, we can't do that because we can't discount if the cost of administration then means that we have to go through extra administration. But anyway, if you buy off the web store, you can use this discount code for December. With that said, let me go forward a little bit faster. I just want to say at ZP, I will be supporting in today's webinar some data that's coming off non-ZP potentiostats. I think with time we will harden up on this a little bit more and actually only support our own potentiostats. But for the time being, at least in this webinar today, I will be talking about or supporting other potentiostats, but I'm in particular supporting those potentiostats because they are using screen printed electrodes from ZP. But just to say, kind of going forward, if people are starting to look for support from ZP regarding electrochemistries, particularly on their potentiostats and also on the screen printed electrodes, then we would really kindly ask that these are on ZP screen printed electrodes and also ZP potentiostats as well. Anyway, a couple of links to ZP screen printed electrodes and a couple of links to ZP potentiostats. With that said, let's go forward. So the questions today, one of the hardest questions to answer is people ask about CGM, continuous glucose monitoring. The thing about CGM, this is a CGM watch. I know some of you who are watching this will already seen this, and this has been talked about. But this watch alone, which is a microneedle watch, the microneedles, you can use them for measuring lactate, hydrogen peroxide, glucose, ketones. You can use them for lots of analytes. But a watch like this, a wearable watch, which is basically breaking the skin and going transdermal, that alone has had 20 million euros invested into it. So when people ask about what's the cost to get to market, they ask us a lot of questions. But I would say, if you've only got a limited budget, use that budget to go to investor meetings. Don't get into the engineering and science of CGM. In academia, it's a different matter. You can solve new problems. But if you're truly a company or an entrepreneur who wants to start a CGM, a continuous glucose monitoring company, with that budget, you may be somewhat better off actually going to investor conferences and finding investors. I only estimate this, but I think Abbott have spent probably $1 billion US getting this device to work. This is the Abbott Freestyle Libre 2. Dexcom, I do have a Dexcom sensor in this office, have spent about $500 million US getting to market, which is fine because now they have quarters that are much bigger than $500 million. But just to say, and then Biolinq in the last 4 years have easily raised $154 million US. So if you are an entrepreneur and you've just raised a few thousand euros, you're possibly better off using the money to help you raise this kind of money, as opposed to coming to ZP and saying, oh well, I want to start a project, because these programs can be quite large. Question number 2 will be about hydrogen peroxide. Question number 3 will be about lactate. Question number 4 will be about gold versus carbon. Somebody was saying for protein detection that we say that gold is better than carbon. What I say is that actually carbon is so much cheaper than gold. Everyone who wants to do a point of care diagnostic or point of need diagnostic, everyone says, oh, I want a point of need diagnostic, it's got to be low cost, easy to use, and so that term low cost is immediately more difficult to achieve when you go for gold. So the only reason I say I prefer carbon over gold when it comes to protein detection is not really a technical reason. It's more important than that. It's commercial. It's very hard. Gold is very expensive. So if you can avoid it in your medical diagnostics, avoid it. Unfortunately, it's the same for platinum as well. You can think about it, people are very interested in platinum. It's got lots of uses. It's got catalytic uses. It's got luxury uses. Gold, has catalytic properties. Is used in the electronics industry a lot. Is also used in the luxury industry a lot. And then carbon. I mean, you don't find the luxury industry super interested in carbon. So if you've got a choice for protein detection, I would choose carbon, and I'll dig into that a little bit more today. Measuring the conductivity of, I say here, of chloroform, it's actually wrong. Somebody's put a photo switch or a photomolecular switch in a chloroform. When the light shines upon it, it will ionize, or it'll form some ions, and then the conductivity will change. So I will dig into that a little bit today. Question number 6, somebody's asking about the stability of screen-printed electrodes. It's probably worth saying that if you watch week after week at ZP, we do talk about screen-printed electrodes a lot. And the thing that I keep emphasizing on ZP screen-printed electrodes is actually not the stability. I think I haven't mentioned stability of screen-printed electrodes in an awfully long time. What I talk about is the repeatability of screen-printed electrodes, because I think that screen-printed electrodes are often used in, we use them in what's called point of care or point of need diagnostics or testing. They're used a lot in glucose monitoring. I've got some glucose strips here. So screen-printed electrodes are famous for being used in single point tests. Now I will talk about the reliability of them in continuous, let's say, monitoring, but it's not something that I super promote. So I will talk about it today, and I'll also comment upon experimental setups. Somebody's got some glucose data. The short answer is I've got a super suspicion that these people have a generation 1 glucose sensor. I think they may be applying a negative potential to those sensors and not 650 millivolts. I will dig into the details, but I can see it in the data. And then something else is just interesting, glucose technologies in general. ZP, we have what's called a barrier layer, and we also have catalytic layers that people can buy as well, enzyme catalytic layers. So 7 and 8 are kind of not so much intertwined, but they are related. Different people, but they're related. Just out of interest, if you are interested in point of care diagnostics or point of need diagnostics, do come to our webinar, 7th of January, 1 in the morning, 1 in the afternoon. It allows the maximum number of people to be able to attend. If you see any kind of links pop up now, just click the links underneath the video and you'll be able to find, or rather the links will be underneath the video. We're also doing a webinar next week on wearable biosensors. So if you're interested in wearables, please come on the on the 12th of December. Again, 1 in the morning, 1 in the afternoon. Academic research, we are very interested in the translation of academic research into products. If you are in academia and you're interested in translation of research into products, please come to that. 10th of December, 1 in the morning, 1 in the afternoon. If you're watching this webinar several months from now, these links will just take you to the most recent or most upcoming webinar. So we do these every month. If you're interested in electrochemical assays, wearable biosensors, point of need diagnostics, maybe one of the best ways is actually to come to a workshop that we're having in Norway. The next one is in January. I will be giving it. It's not a lecture type workshop. There'll be content, and you can have that and ask questions. There'll also be a lot of lab testing as well. So if that's of interest, then that link will be underneath this video as well shortly. Just about CGM, continuous glucose monitoring development. I know I think I'm boring myself with how much I talk about this on this in these webinars, so I apologize for it. But let me answer these questions. ZP is fully capable of prototyping wearable biosensors. I would typically say, just to kind of give people a sense, in order to kind of produce maybe a first prototype in a wearable application, then I would budget something like £50,000 or €50,000 or $50,000, whatever your budget is. Now, what ZP can actually prototype much more efficiently than most companies, or most especially most startups, because we actually have a a lot of the kind of hardware that's necessary for this kind of thing already in place. So we do have electronics perfectly capable of running CGM, continuous glucose monitoring, continuous lactate monitoring, continuous hydroperoxide monitoring. These technologies are not available on our web store. We will only use them in helping an actual client who's opened a budget with us. By opening a budget with us, it allows us to have a more detailed conversation. Then we can come up with a plan, let's say, and then we can actually act upon it. But the quick answer is ZP is more than capable of producing prototypes. Costs and timelines. I think the first prototype with ZP, or with anyone who's really knows what they're doing, should be about $50,000. And we're more than fully capable of assembling them. You'll see some pictures in a minute that will kind of give you a sense of what we do. We're more than capable of mass production. Mass production, though, in the end, really depends on what comes out of the development. So the whole term, can you mass produce? Yeah, sure, I can mass produce. In fact, I'll reach down here now. Let me just grab. Here's an example of mass production. So the answer to mass production is simple enough. But of course, somebody might have, what comes out of a CGM project could be very specific. So that there's a very nuanced that. But in order to kind of engage with these kind of conversations, people really do have to have serious budgets in place. We are at ZP, we are ISO 13485. So we do the contract development, contract manufacturing of medical devices and medical diagnostics. And we definitely have people on the market with true medical devices, and we've got them through regulatory. And of course, we can customize. In fact, in the end, anyone who's doing, let's say, a wearable, including continuous glucose monitoring, you have to do some kind of customization. I mean, an obvious thing would be, if you tried to do something like this, then you would be infringing their patent. So you have to do customizations by definition. And by the way, CGM, continuous glucose monitoring, is really a litigious industry. So you absolutely have to do customization. Otherwise, you would just be doing what's called a Me Too. If you do a Me Too, you're open to litigation, and the incumbents on the market are not going to be happy with you, and you'll find yourself in litigation. It's probably worth saying that ZP goes beyond just prototypes. And at some point you have to do animal studies or on human studies, and we're more than capable of doing that. These are our images of tests that we are doing with clients. So the quick answer is, if you've only got a very limited budget and you're interested in the CGM space, use the budget to go to investor conferences, use the budget to pay for hotels, use it for flights. You need to be well funded in order to do this work. If it's in academia, that's a different matter. You can spend, let's say, years getting there. But if it's industrial, then time is money, and you have to get there quick. So I would not rather, if it's a limited budget, spend it more on finding investors rather than getting into an engineering program. Question number 2 about hydrogen peroxide sensors. Just out of interest, so hydroperoxide detection. In fact, ZP does have a client on the market doing hydroperoxide detection. They're doing hydroperoxide detection for inflammation of the lungs. They're doing it in COPD, chronic obstructive pulmonary disease. So ZP, it just occurred to me now, I was wondering how I was going to answer this question. And it just occurred to me that ZP does have a customer on the market. In fact, I'll just grab their device now. They're called Exhalation Health. This is the device, or this is at least a disposable, and it's for measuring COPD, chronic obstructive pulmonary disease. I can talk about this quite openly because it's in the public domain. You'll also find that my name is on a lot of their patents as well, so you may be able to find them that way. So the quick answer is there are people measuring hydroperoxide in the medical space. In that particular case, they're measuring lung inflammation, which might be interesting. To deliver a fully assembled prototype, for example, I think and it is prototype, and I'm going to sort of say TRL3, technology readiness level number 3, because I would say that's at least $50,000. I know because that's what we're doing at the moment for various clients. In fact, I'll use the answer number 1 to answer number 2. So in answer number 2, somebody is using hydrogen peroxide on the breath as an inflammatory marker for COPD, chronic obstructive pulmonary disease. for helping to monitor and control those kind of patients. You can use microneedles potentially. You'll be the first ones to do it. But I would say this, right? So the question here is, can you use microneedles to monitor hydroperoxide? And the answer is yes. And the reason being that is because glucose oxidase, lactate oxidase, they're actually converting lactate plus oxygen into hydroperoxide. Glucose oxidase is converting glucose plus oxygen into hydroperoxide. So in fact, all these sensors are actually hydroperoxide sensors already. It's just there's an enzyme between the analyte of interest, and that converts the analyte of interest actually in from oxygen into hydrogen peroxide, and we're detecting it. So hydrogen peroxide as a wearable sensor already has traction, let's say. Most people just don't realize it. In terms of microneedles, I would suggest that the people who are in the public domain leading is a strong word, at least in the public domain with the highest profile, would be Biolinq. So Biolinq are probably doing what's called a generation 1 glucose sensor. So in fact, on their microneedles, they will have glucose oxidase. The glucose oxidase will react with the oxygen and glucose and generate hydrogen peroxide. So there are people using microneedles to effectively measure hydrogen peroxide. It's just not so apparent because of the biochemistry, but in fact, that's what's going on. ZP does have a lot of tools. We sort of help. If a company's quite serious about doing a wearable biosensor, then ZP won't support them through bits and bobs off the website. We will only support if you engage us in serious programs, because otherwise people will sort of buy bits and bobs and then they want, you know, some support and all this kind of stuff, and we're like, no. If you're going to do it and you are a company and you look fundable, we would say do a program with us, because then we know we can control all the variables. We don't have to necessarily so much teach. We can show, but otherwise people are expecting us to be able to teach them and support them while they come up the curve, and it's like, no, we're in a rush to get people to market. So we would rather not be selling bits and bobs to startups. If startups want to do wearable sensors, open up a budget, engage with us, even come on site with us. And in fact, watch for one of the questions in a minute, so I'll describe that. So we would basically sort of, and if somebody said to me, okay, I'll open up a budget with you, I'd say, okay, give us about $50,000 and we'll bring you to technology readiness level number 3. So we are looking, we're happy to work with well-funded startups, and we don't really mind what the configuration is in terms of wearables, transdermal, microneedles, not necessarily sweat sensing. When I say that is we are doing sweat sensing for lactate. And also I'm not going to suggest implantables actually, but we are doing implantables with people as well. It's just to say on lactate sensing, for example, we do have some well-funded US company coming next week. And in fact, we will be doing on-person testing on ZP lactate sensors to help them scope out new business opportunities. So we do like companies that are well funded, companies that are dynamic, companies that can move fast, and if it's things like lactate and are kind of exciting for us, so we're quite happy to have people come, but there has to be a budget in place in order to afford all the engineering and stuff. But watch us on LinkedIn or watch us on YouTube, because you will see some images coming out of this pretty soon, and maybe some videos as well. Somebody did send me a question. This is now moving on to protein detection. I probably did say carbon is better than gold, but they think that the answer that I'm trying to give them is because it's technically better than gold when it comes to protein detection. It's only cost is the only reason I say this. This is a gold electrode from ZP. We try to keep our screen printed electrodes at a reasonable price, but what I'm saying here is, it's 479 euros for 50 sensors. So, you could say, wow, that's expensive. It's the gold. And actually, the last few years with inflation, and sometimes people are looking for safe havens for their money, gold has gone up in price. Gold is an expensive material. It's also, something that people invest with. It's something that people use as a luxury item. It's not good to get gold into your supply chain, because you're always going to compete with lots of other people who want gold for various applications. So gold is not a good first choice for anyone who really wants to make a low-cost biosensor. So that's my kind of, let's say, comment on gold. Now look at carbon in the meantime. Here's a carbon screen printed electrode from ZP. We sell at less cost. We sell twice as many sensors. Essentially, the pack size here is 100, as opposed to pack size previously being 50. And this is on one of our kind of sensors in a cartridge format. So I'll just hold up what I mean by sensors in a cartridge format. This is sensors in a cartridge format. You know, so there's a lot of value being put into this sensor, the way we kind of, you know, have put it into a holdable system, the way it just clicks in and out of these kind of potentiostats that I have in front of me. So we've added a lot of value to that. So quick answer is, the reason I sometimes say that carbon is better than gold is because gold is expensive. Gold is a sort of luxury item that people would put into jewelry. Carbon's not necessarily something that people would so much put into jewelry. Now, and it's easy enough to functionalize carbon, for example, with antibodies, and antibodies are often used for proteins. You can use something called pyrene NHS, which the pyrene group sticks to the graphite. The NHS group is quite reactive. You can react the NHS group with antibodies, and then you functionalize your surface. You can block for a non-specific binding with a material like chitosan, and you've made yourself a biosensor. And then you can follow that process using techniques like cyclovoltammetry. You know, so the functionalizations can all be kind of confirmed by using something like cyclovoltammetry, using a reporting molecule like ferricyanide. So quite straightforward. And then you can actually read it using something like differential, I was going to say differential pulse voltammetry, but I actually prefer square wave voltammetry, which gives me an opportunity actually to promote this particular potentiostat that I have in front of me. This is the Sensi Oil potentiostat from ZP. As I say, we are much more keen to support people on this potentiostat, but it does have, it's driven by QR codes, so I'll probably be doing some work in a minute. Amperometry, open circuit potentiometry, impedance spectroscopy, linear sweep voltammetry, cyclovoltammetry, and square wave voltammetry. So it's quite nice because actually we can follow the functionalization of these screen printed electrodes using cyclovoltammetry, and then we can actually read the assay using square wave voltammetry. So if you're looking to get into this field and you're definitely looking for support from ZP, then we would ask you to, obviously, it makes our lives easier if you're using the same instruments as us. Because what's nice is I'll actually be answering some questions in a bit, and I know exactly what's going on because they're using our potentiostat, so thank you to those people. But that said, the reason I say carbon is better than gold is not because I'm trying to say it's better technically, though I do think it actually has advantages. It's because gold is expensive, and I don't even have to justify that statement. I think people would understand that gold is expensive for all the reasons that society wants to use gold. Now, this is an interesting question. So somebody wants to measure that. I say here the conductivity of chloroform, and that's slightly miswritten. They have an organic molecule that's in chloroform. They're going to illuminate it with some light, and in doing so, the organic molecule is like a photo switch. It ionizes and changes the conductivity of chloroform. And chloroform is an organic solvent, so I can absolutely understand that chloroform gives them a bit of a problem because it will chemically attack lots of screen printed electrodes. At ZP, we actually do have a range of screen printed electrodes that are very organic solvent resistant. And I just discovered, I should have realized this, but I didn't initially realize that, in fact, this potentiostat from ZP can actually take these solvent resistant screen printed electrodes, really no problem. So I was a bit, I did test it just now and I was like, oh, okay, yeah, they actually do fit. Anyway, with that said, let's talk about the problem, or let's talk about the science. So we've got this chloroform. Chloroform is quite an aggressive organic solvent, so I can understand their problems and concerns. In it, they've got a photo switch, which when they illuminate it, it probably ionizes, and in ionizing, it probably changes the conductivity of the chloroform. And at ZP, we just did some recent tests actually, and we do have screen printed electrodes that are resistant to chloroform, but we have lots of screen printed electrodes that are not. So I will be pointing out the ones that are. if anyone's interested, there's a long time video up on YouTube actually where we do show us testing some of our screen printed electrodes in organic solvents. Once upon a time, we used to promote these electrodes quite heavily, but they've got more and more expensive over the years as the things like the gold has got more expensive. So we don't promote them so much these days, but we do have these things. So this old video might be useful if you haven't already seen it. And also, if people go forward with these screen printed electrodes, also know that they do work on our sensi or potentiostat. And I did make sure that everything fitted by having a little test myself earlier on. So unfortunately, we only do large pack sizes. Now I show the spiral electrode here because the spiral electrode would not most, but a lot of people would say, a spiral electrode's a bit like an interdigitated electrode. Interdigitated just means you've got electrodes that kind of do this, where you've got these kind of fingers that interdigitate. But we only do large pack sizes, and we just can't do small pack sizes anymore. We have to do large manufacturing runs, so they can be quite expensive. But we do have a smaller pack size, which are solvent resistant. So this, and I realize I don't even have a link to it, but these are the electrodes that are resistant to organic solvents, and these ones are the ones I would recommend for this particular work. What else can I say on this? Yeah, there was something I wanted to say, which is people are very concerned about the geometry when it comes to making conductivity measurements, but we've done a lot of work over the time, and we find out actually the geometry, at least on this scale of screen printed electrodes, makes very little difference to the conductivity that you can otherwise measure with these electrodes. So we find these perfectly good for doing conductivity measurements. Now this is a long, hard question. Okay, so at ZP we try to make a really reproducible screen printed electrode, and reproducible. And today I'm going to talk about stability. So let me just jump into this a little bit more and say, what are we trying to do at Z, or what are they trying to do? So somebody's trying to use, and I appreciate them doing this, by the way, because they're actually using a ZP screen printed electrode, and they're trying to make a sensor from it. So thank you very much. It's appreciated. They will be modifying our screen printed electrode. Now this is important. If you're going to modify a ZP screen printed electrode, it's the modified electrode that you should be assessing. It's not actually assessing our screen printed electrode as comes, because that's not the product that you're trying to make. So I will talk a lot now about how to measure and how to improve the stability or the experimental setup, let's say. But in the end, you'll see me finishing with, but it's actually not the screen printed electrode you should be testing. You should be modifying it and testing that. But let me first of all just make a philosophical discussion. I say here demo. So let me just go really quickly to do a quick demonstration. So let me grab an archetypal screen printed electrode, which I will do. Let me put it into a meter like this. Let me get a sample like this. Ow. Right, let me get the blood. Right, now the reason I just did all that is because I want to make you realize that ZP, the parameter that we work on, so that's my blood glucose, 89 milligrams per dl, which is a bit low. 90 milligrams per dl is kind of, well, it's only bits. It's within experimental error, or the error of this machine, that's probably, you know, that's exactly on. Now, that is the thing that ZP is working on when it really comes to screen printed electrodes. Well, what I mean by that, I mean these kind of technologies can be low cost, they can be reliable, they can do fast assays, but it's one sample on there and you get a result. That's what we're focusing on at ZP. In fact, I'll give you another demonstration. In fact, I will talk about the stability of the question, but understand that stability of screen printed electrodes is not actually what we're promoting. If you watch a lot of our materials online, I always think that screen printed electrodes play a really good role in point of care, point of need diagnostics. I really do think they play a great role. So here comes another ZP. This is a ZP product. Put the sample on there. Hit continue. Hit start. And off it goes. That is what ZP uses screen printed electrodes for. Now we're also using them in some client projects for continuous monitoring. But I just want you to know that that's why we have screen printed electrodes. It's for this kind of work where you have a sample, you put it on there, you get a result. So now I'm going to start talking about people who are measuring things over 3 days. I'm actually going to show you data for 40 days. But I just want people to realize that we promote screen printed electrodes as a point of need, point of care diagnostic technology, not necessarily as a platform for people doing several days of monitoring. That's not a parameter that I often talk about on this channel, and in fact I'll show you what I often talk about on this channel. On this channel we will often talk about, hey, here's a screen printed electrodes, and hey, here's us doing it in a point of need measurement technology. So we do talk about screen printed electrodes, but what we're actually talking about is reliability, and that's reliability or repeatability, I should say. But what we're interested in is actually, are all these electrodes similar? And are they similar that we can use them in a medical diagnostic? Now, our metric for similar is cyclovoltammetry. So we're only answering that question by cyclovoltammetry. So once people start bringing out different techniques, we have not tested these electrodes, for example, similarity by OCP or by EIS. No, we have electrochemically ensured similarity or repeatability of manufacturing, but it's by discrete, fairly fast cyclovoltammetry experiments. And this is something that I'll talk a lot about, and these are old slides, but I just want to re-emphasize that what we're doing is demonstrating repeatability, and this is something that I'm not going to say now because we do have lots of videos about it, but we do really check the repeatability of our screen printed electrodes. And these tests are done quite quickly, and we're making sure that all those electrodes are repeatable. But what we're not suggesting is that they will work in your application over several days. Now, I will show you several days worth of data, but just to kind of say that it's a philosophical thing that, sometimes when people ask me questions and I say, oh yeah, screen printed electrodes are good quality, I am definitely thinking about this type of application, the blood glucose type application, or the quick chili test I just did. I'm not necessarily thinking about some long-term testing in water. So we do have great repeatability, and we are able to make really good assays out of them. Now I'm going to dive into the actual, let's say, question itself. So somebody's taken some pristine screen ZP electrodes. We do appreciate them using our electrodes. They're connecting them to an adapter. And they're using a third-party potentiostat. As I say, at ZP, we will support, for the time being, potentiostats, but we are going to be getting more and more keen. As we increase our range of potentiostats, we'll get more and more keen on supporting our own potentiostats. But you can see I'm supporting somebody else's potentiostat here. And they've been immersing our ZP screen printed electrode in solution. And they've been collecting OCP, open circuit potentiometry, over several days. And they'll give me the details, and I will comment upon that. Now, what's really good about this, and I appreciate these people doing this, they gave me a very detailed presentation. So it's very hard when people say, I'm having a problem with your technology. But when you give me a set of slides, you will find that we will really analyze things for people. And they gave me photos as well. So big thumbs up and thank you so much. It really makes my life a lot easier. This is an old image that is on the website that I put up there in 2019, and I'll link to it in a minute. But we know that when people are doing open circuit potentiometry and they're wanting to make biosensors, they would love the signal to be flat line like this. Now, in reality, you get this. Now, I've been making these statements since 2019. So there's a perfect and there's a reality. So I've always been quite open about, in particular, open circuit potentiometry can be quite a sensitive technique to not just the analyte of interest, but the matrix, the sample in which you're testing. And I think some of the problems this person, I say problems, I think their signals are well within my expectations, but I will talk that I think their solutions are not quite as stable as they would otherwise think. So I appreciate them giving me all this data. Now, when I look at this data, they're showing me data here over 3 days. So thank you for giving me the data. Thank you for giving me such a nice presentation on it. And then it allows me to analyze your data. So I look at this and I think, okay, so you've got something like a 27 millivolt change over approximately, 3 days, let's say, or 1 day, 2 days, let's say. This is pretty good for me. Now I can understand that if this was pH, a voltage change of 27 millivolts over 2 days, 27 millivolts is equivalent to like 0.5 change in pH. So you went from 7 to 7.5, or you went from 7 to 6.5. So I can understand that you'd want that stable. But if you look at OCP sensors like pH sensors, every time you walk up to an OCP sensor in the lab, call it a pH sensor, the first thing you notice is there's a couple of calibration solutions there. So it tells you that OCP sensors, open circuit potentiometry sensors, which pH is the sort of biggest measurement used in the world using OCP, they drift, and that's why those calibration solutions are in place. So I would not want people to think that we were suggesting that a screen printed electrode from ZP is going to be rock solid in an OCP measurement over 3 days. Now you will see me show you some really good data, but we had to work really hard at that, and I'll try and give you some clues about how we actually went about that. But thank you so much for giving me the data. It does allow me to answer your questions in more detail, including things like I can see that you got 27 millivolt change over approximately 2 to 3 days. It's actually quite good. And if you say, well, no, I need it to be more stable than that. Well, think how long people have been making pH sensors and pH meters for, and there's still calibration solutions next to them. OCP is sensitive to drift. No matter whether you're using screen printed electrodes or glass spore electrodes from Mettler Toledo, for example, they drift. Just out of interest, I did see this data here as well, where you kind of, it changes quite dramatically, let's say. And I just want to say that we've also got that kind of data, and I'm also going to give you a link to it. But when we got that kind of data, what happened was we were using a beaker that was open, and we were actually getting evaporation from the beaker. So in fact, the solution was changing its concentration. And when I look at your experimental setup, I'm certainly not critical of these people, because they know how to present their science, they know how to tell me the story, makes my life easier, including the good quality images. But the beaker is it's open, there's no temperature control, there's no temperature logging here. So I can see a few things, which is it's not necessarily sealed. You will get evaporation over those course of those days. You also got the possibility of exchanging gases with the atmosphere as well. OCP is a sensitive measurement. If there's gas exchange between the environment and your solution, which there definitely is, then it can show up in the OCP measurement. Also, you're not necessarily monitoring or controlling on temperature, which is fine, but OCP can be sensitive to temperature as well. As I say, I wasn't actually that disappointed with your data. You are, but I'm not. But that's because I would expect this kind of change. Now, what I would say as well is that you're using our carbon electrode. You've given us nothing specific to do. So what I mean by that is, when you turn this carbon electrode into a biosensor, then monitor whether your now made sensor is actually stable or not. But carbon, it's almost like it's very undefined what it's actually doing in this situation. It's just sitting in a solution, and it's reacting to whatever the solution's up to. So it's not necessarily the carbon that's changing. It's actually just reacting to the solution that genuinely is stable. It's also probably worth just knowing that if this is sitting in light for several days, silver chloride, most silver and silver ions are light sensitive, so just be aware of that. What you need to do is actually functionalize that electrode and then do your assessment. But Carbon doesn't know what to do. You haven't given it any specificity. Once you put your special materials on there, then you assess it. But understand that the carbon is only there, provided by us in your experiment, to detect your, essentially your potential. You have to functionalize it. So be careful, because you're myopically going in on the carbon, but that's not the material. Actually, by the time you use this carbon electrode, let's say, it will already be sitting underneath your material. So you will have built specificity. So you really need to be assessing the stability of the sensor that you make, not the underlying material at the moment. But at ZP, we have done this kind of work in the past. And in fact, we did manage to get some very stable data. But what we did is we were in a Faraday cage. So we made sure that there was nothing, no stray sort of electromagnetic interference on our experiments. We sealed it. So we weren't exchanging gases, we weren't allowing evaporation to take place, no matter how small, and we also monitored the temperature in it. So we really had to put a lot of work into ensuring that we could actually get a, let's say, a stable signal. But I cannot promise you a stable signal. It's not something that we test for. What we do is we make a screen printed electrode that we test the repeatability on, because we expect people to use them in a point of need diagnostic. By the way, we do use them in continuous work. In fact, this is 40 days worth of work, but it's quite a rare occurrence for us, and it's not something that you even will very, very rarely actually hear me even talking about it. But you have to make sure that solution is really isolated. It's not evaporating, that you're using Faraday cages, that you're at least understanding the temperature swings in that laboratory. There's all sorts of parameters that you have to measure or you have to control. Now, this is an interesting one. I'm changing direction slightly here. Now I'm going to talk about glucose data. So they are using our instrument. So this is my favorite one to talk about, because they're using our instrument, they're using our glucose sensors, and because of all that, it makes it very easy for me to support them. One thing I didn't say in the previous question was, because I don't really know if that potential stat is drifting either. I doubt it, but just FYI. They're using a generation 1 glucose sensor. It's interesting actually. They said they're following the instructions on the sensor all for glucose sensing, 30 microliters. I wonder actually. I wonder. I think I might have realized what's going on here. These people probably need to measure amperometry. They've got a generation 1 glucose sensor. They're following maybe our glucose protocol, and I realized that in fact they need to be running this sensor in an amperometric mode. So I think I might understand actually what's going on here now. There's a bit of a confusion here. We do have glucose sensors that go with the sensor all, and we also have glucose sensors that will run upon it. But I think these people actually need to be running their sensor all in an amperometric mode. So I'm just going to show my screen. I think they actually need to be, they need to actually be coming into the amperometry mode, and then they need to be setting the applied potential to about 650 millivolts, as opposed to, I can see what they're doing. They're using this. It's not their fault. I can see they're using the QR code that the machine has. Just getting this to 650. Yeah. They're using the QR code here, which is for glucose, when in fact I would want them to maybe use the amperometry mode here. So it's probably slightly our fault. I see what's going on here. We have people that use glucose in the food industry, but these people are not in the food industry. They're more in the sort of science industry. So in fact, they need to use this QR code here. So first of all, I think you should be running this in amperometry mode. I think you should be scanning the QR code for amperometry mode. I think you should be running for 650 millivolts. I've got a suspicion that you're not running 650 millivolts, and I will talk about that now. This is what their data looks like. It's not in the, well, sorry, and I say that and I realize that I'm sharing the wrong screen, so let me just back up slightly. Which is making me think I need to be here, and so they're using a glucose sensor. I think they should be applying 650 millivolts to this sensor. but I realized that they may be using a QR code for the glucose sensor when actually I would want them to be using the amperometry mode on the potentiostat. So that's on me to kind of get them and get that corrected, let's say. Their signals are showing a reduction signal when really they should be, with those generation 1 glucose sensors, using a potential of about 650 millivolts so that they actually get an oxidation signal. So I can see all their signals in a reduction mode, which doesn't make, first of all, it confused me, but now it doesn't confuse me because I realize that maybe they're using the QR code wrong, not wrongly, but I need to just correct, which is what I'm doing in this webinar. But the quick answer is, I would want your data to actually look. So it's probably worth saying that when we get our data like this, we're actually doing it in a beaker, you're doing it in a drop format. That's not the big, let's say, concern here. But the way these glucose sensors work, and you could also describe lactate sensors and hydrogen peroxide sensors, but in the end, glucose reacts with oxygen, forms hydrogen peroxide, and we want to detect that hydrogen peroxide, and we use a potential of 650 millivolts. If you're using 650 millivolts, then your data will look like this, but your data looks like this. And it may not be your fault. It may be because they've scanned the QR code for glucose, when actually I'd want them to scan the QR code for amperometry, and then I would want them to manually put in 650 millivolts. So that's probably, so the data's going in the wrong direction. It's easy for me to fix because you've sent me a well presented, detailed slide deck. It really helps me. But I'm expecting your data to kind of come down like this, when in fact your data does this, which confused me. But scan the QR code for amperometry, set it for, set your data for 90 seconds if you want. But the potential that you're meant to be applying is 650 millivolts, then send that to me. And that will really help me figure out what's going on here. Last one. Is the barrier layer that we sell on our website the same one that we use on our glucose sensors? The quick answer is yes. We do sell platinum screen printed electrodes. So people can make their own glucose sensors on top of those platinum screen printed electrodes. I don't believe we make a platinum microneedle anymore. Just out of interest, gold microneedles can also be used to make CGMs as well. We do sell some gold microneedles. If you're a well-funded company, don't go by the gold microneedle route. This is the route, well, let's say gold microneedle route. Don't buy gold microneedles off our website, because if you're a company, I want you to basically engage with ZP more deeply. With academia, the timelines are longer, the budgets are smaller, then people can engage with our technology through, let's say, buying stuff off the website. And we don't do a platinum microneedle, we only do a gold microneedle, but you can use gold also for the detection of hydrogen peroxide, so it will work with these solutions. The shelf life of these different solutions, I did look, and I must admit, this barrier layer, this polyurethane, in fact, it lasts for years, but I do agree that we don't actually have it in the data sheet. So this is not an official statement from ZP, but we do use it for years, but we do keep it in a cool, dark cupboard. I didn't say fridge, I did say cool, dark cupboard in an air-conditioned, let's say, room, so that, we're not getting extreme swings of temperature, but it does last for years. Your other one is about the glucose sensing activation solution. With that one, it's a hard one for me, but I did notice that, in fact, it does say store at minus 20 degrees Celsius on the data sheet. So I did find the data sheet and I read it, and it says minus 20 degrees C. So that really helps me, because if it's minus 20 degrees C, then basically it's frozen. And that's what will help you have a prolonged shelf life. So I'd hope glucose oxidase solutions for us can last a long time, but only if we keep them at minus 20 degrees C, not 5 degrees C, not room temperature, but minus 20 degrees C. Then they can last several months in the freezer like that. So question number one, if you've got a limited budget and you are really trying to make a commercially available CGM, continuous glucose monitor, and you've got a limited budget, use the budget for finding investors, because otherwise you'll just do a little bit of work, you'll run out of money, and it takes, a lot of money to make a CGM. Question number two, hydrogen peroxide sensors. Hydrogen peroxide, I think, is quite a doable analyte. ZP does have a customer actually on the market measuring hydrogen peroxide actually on the breath, which I would suggest is quite a hard thing to do. They're called Exhalation Technologies. They are CE marked. It is used in hospitals. It's allowed to be used with patients. And they're using it as an inflammatory marker for COPD, chronic obstructive pulmonary disease. Lactate, follow us on LinkedIn, follow us on YouTube. You're going to see that a lot of wearable work popping up on Lactate soon enough. I don't say carbon is better than gold. I just say low cost and gold do not go together. Carbon's a perfectly good solution. We use it a lot for protein detection. The conductivity of chloroform is only tough because in fact the materials are quite expensive from ZP, but I have linked to a pack of 40 sensors. That is the best recommendation I can make. Stability of screen printed electrodes. Turn the screen printed electrodes into your sensor and then judge your sensor. If you want to do repeatability tests, don't allow evaporation, don't let gas exchange, and also ask yourself the question, is what I'm trying to achieve reasonable? And look at a pH meter in the lab and ask yourself, how often or how many times a day does somebody recalibrate that device? And then you can think, well, why do we keep recalibrating it? It's because OCP, open circuit potentiometry, is a technique that is prone to drift. With the glucose data, I think the potential that's been applied to this generation 1 glucose sensors, it's a reduction current that you're showing me. I think it means that there's a reduction potential being applied, so I think that needs to be changed to 650 millivolts. If I'm making a mistake, I apologize and we'll sort it out. Glucose technologies, the polyurethane barrier layer will last for years. Just keep it in an air-conditioned dark room. The glucose oxidase solutions, it does say -20, and that really helps. So it'll last several months because basically it's frozen. And also glucose oxidase itself is quite a robust enzyme. So I'd say thank you here. I'm sorry if I've any ranting that's gone on today. I did the webinar before I had my lunch, so hungry. But if you've got any questions of ZP, please don't hesitate to reach out to us. Okay, thanks very much.